Good afternoon, everyone. I P. Kirtisika, Assistant Professor of Department of Pharmaceutical Analysis from Sri Padmavati School of Pharmacy, take this opportunity to extend a hearty welcome to all the virtual audience. So we came to the end of this Indo-African webinar series. So in the past days on September 29th and 30th, we have seen the two sessions based on this role of pharmaceutical analysis and quality assurance in the vaccine development by Noor Ahmad Syed Geta sir, as well as role of biotechnology and microbiology in this vaccine development by Mohammad Rafi Mokla sir. So today is the third part, which is based on the theme challenges and opportunities in drug discovery in the developing countries, an example of cutaneous leishmaniasis by our honorable speaker, Abdrrahman Shemsu Surur sir. So, in this topic, in this today's session, you are going to have a glance of what are the various opportunities and challenges in the drug discovery. So, before starting the session, I would like to invite all the dignitaries to this program. First, I would like to invite the coordinator of today's program, Dr. A. Srinivas Charan sir.
Thank you.
Welcome. Now it's my honor to invite the organizing secretaries of this today's program, Dr. K. Rashekar sir, HOD of the Department of Pharmaceutical Analysis. Welcome you, sir.
Thank you.
It's my privilege to invite the convener and principal of Sri Padmavati School of Pharmacy, Sri Dr. D. Rangnaiklu sir, to this program.
Thank you. Namaste.
Welcome you, sir. Now, it's an honor to invite our today's speaker, Dr. Abdrrahman Shemsu Surur sir, MPharm PhD, Postdoctoral Research Associate, Center for Innovative Drug Development and Therapeutic Trials for Africa, Addis Ababa University, Ethiopia, to this session. We welcome you, sir.
Thank you for having me.
Thank
Yeah,
you.
welcome, sir.
With the best wishes of Chairperson of Sri Padmavati Educational Institution, Srimati P. Slochna Madam, and Director of Sri Padmavati Educational Institution, Sri P. Praneeth sir, will start the session. So let us begin this session with the invocation song.
वक्रतुण्ड महाकाय सूर्यकोटि समप्रभ निर्विघ्नं कुरु मे देव सर्वकार्येषु सर्वदा.
Thank you. We all feel blessed. Now, before going to see the session, so I would like to present a profile of our esteemed institution, Sri Padmavati School of Pharmacy, to the virtual audience. Sri Padmavati School of Pharmacy was established in the year 1997, so by the founder, late Dr. P. Mohan Rao sir. Now under the leadership of Srimati P. Sulochna Madam and Sri P. Praneeth sir, Director, the institution has been offering the different courses like B Pharmacy, M Pharmacy, PharmD and PharmD Postbachelorate, and the postgraduate courses like Pharmacology, Pharmaceutics, Pharmaceutical Analysis and Pharmacy Practice. SPSP always stood first in achieving many awards like Campus Preparedness Award, which was received in the year 2015, and it was a recipient of 8 Pratibha Awards and 1 University Gold Medal. And I'm really proud to say that we have 150 plus international and national publications, and our faculty and students has presented more than 250 presentations at the national and international conferences. And our institute has 8 PhD awarded faculty. Every year, minimum 10 plus students were qualified in the competitive exams like GPAT as well as NIPER. And our college was very active in conducting the regular workshops and seminars every year. And these are the list of research scholars who are pursuing their PhD at Sri Padmavati School of Pharmacy. So SPSP always stood first in encouraging all the scholars and providing the facilities to do their research works. And this is about the alumni. So SPSP has a lot of history of alumni, which has settled in different positions like government positions like drug inspectors, research officers, central and state pharmacists, as well as in academics, university and college professors, associate professors and assistant professors. and also in hospital sector, industry sector, as well as in entrepreneurship. These are some of the activities which were conducted at the college, like National Pharmacy Week celebrations, World Pharmacist Day celebrations every year will be conducted at the campus, and many competitions will be conducted, and rally will be conducted, and the social welfare activities. And we have extracurricular activities conducted by different clubs like dance and music club, sports and games club. We have career guidance cell, placement cell for encouraging the students, and grievance cell as well as anti-ragging cell. And every year, SPSC always stood first in conducting the annual day celebrations, Pharma Week celebrations, social welfare activities. And now coming to the Department of Pharmaceutical Analysis. So now we are at the decennial celebrations of the Department of Pharmaceutical Analysis. The department was established in the year 2010, and our department have experienced faculty, and we have sophisticated instruments like HPLC, FTIR, UV visible spectrophotometers, and many of the novel project and research works has been carried out in the Department of Analysis. And we also provide analytical testing services for outside students as well as organizations. The department has conducted many workshops and seminars for the upgradation of knowledge of students, and we have peer reviewed in journals in most focused industry and highly reputed journals. And the achievements of the department is we have done the advanced research works on QBD, chemometrics, as well as analytical method development. Every year, more than 95% results in the Department of Analysis, and 100% placement for the passed out, and well-settled alumni in various domains like industry, teaching, government services, and banking services, etc. So these are the alumni of the Department of Pharmaceutical Analysis who are well settled in the pharmaceutical industries like Arabindo, Microlabs, Dr. Reddy's, Divi's Laboratories, Laurus Labs. So all the students have been well settled in many of the industries as well as in the teaching sectors. And every year we'll conduct the alumni meet and the industrial tip to the students. So thank you one and all. This is the brief profile of our esteemed institution, Sri Padmavati School of Pharmacy. Now, I would like to invite the convener and principal of Sri Padmavati School of Pharmacy, Sri Dr. D. Ranganaiku sir, to present the opening remarks for the today's session.
Thank you, Kirtishika. Indeed, it's a great pleasure to associate with you all on this Indo-African webinar program, 3-day series program. It's happened to be third part today. So, the respect of our chairperson, madam, and due respect with our director, sir, and today our guest, Dr. Abdrrahman Shemsu, and all delegates who have registered for this today program, and all my dear students and faculty, especially is it audible?
Yes, sir. It is audible, sir. Yes, sir.
Okay. The program has been designed and with a kind of intention to upgrade the knowledge, the global wise, especially pharma people looking at the crisis of this pandemic situation. We noted it's in a kind of opportunity where we can have the kind of gathering of all virtual candidates, especially to update this scientific innovations. This reason we found this kind of opportunity. So kind enough, all the in collaboration with the Indo-African people has been arrived here, and today happened to be one of our resource person, Dr. Abdrrahman Shemsu Surur. It's a very pleasure to invite on behalf of all the management and director and our students to take care of this today's section. Of course, no, this session definitely is going to be enrich the minds of all research fellows regarding this the development and the challenges and development for the drug discovery, especially the organism that's named cutaneous Leishmaniasis disorders. And there's very crisis in the what I noticed in the African area. There's a kind of pathogenic invasions, kind of disorders. It's very useful program for development of the discovery and innovation program. Definitely, this session is going to be very much useful for entire global wise for all the industrial pharmacists as well as the clinical pharmacists to innovate to safeguard the healthcare system. So with this, I invite Abdrrahman Shemsu Surur to take the lead today's section. Thank you very much, Shika.
Thank you very much, sir, for your valuable address. Yes, yes. Thank you very much, sir, for your valuable address. Now, I would like to invite Srimati C. Harita, Assistant Professor of Department of Pharmaceutical Chemistry, to present the brief profile about today's speaker, Dr. Abdrrahman Shemsu Surur, sir.
Thank you, madam. Good afternoon, everyone. And it's my privilege to introduce our today's speaker, Dr. Abdrrahman Shemsu Surur Garu. MPharm PhD Postdoctoral Research Associate, a World Bank Center of Excellence for Innovative Drug Development and Therapeutic Trials for Africa, Addis Ababa University, Ethiopia. Sir did his bachelor's degree in pharmacy at Mekelle University, Ethiopia. Sir completed Master of Science in Medicinal and Pharmaceutical Chemistry at Addis Ababa University, Ethiopia, with the thesis of analysis of 3-methyl-4-nitrophenol, a major metabolite of phenyltrethione in mice urine using HPLC method. Sir completed doctorate in Medicinal and Pharmaceutical Chemistry at University of Greifswald, Germany, with the thesis of profiling the activity and hepatotoxicity of flupirtine through medicinal chemistry approaches. Sir has more than 10 years of teaching experience in various positions like Lecturer of Medicinal and Pharmaceutical Chemistry at University of Gondar, Ethiopia from 2008 to 2016. Sir worked as doctoral student in Medicinal and Pharmaceutical Chemistry at University of Greifswald, Germany from 2016 to 2019. Sir is currently working as Postdoctoral Research Associate at Addis Ababa University, Ethiopia from 2019. And coming to the achievements, Sir has more than 2 years of experience as the head of the department, University of Gondar. Sir worked as secretary and later as chairperson of Woreda for Youth Library for 2 years. Sir worked as vice president of the Graduation Bulletin Committee. Sir has more than 10 publications in internationally reputed peer-reviewed journals and also participated and presented in scientific conferences. Sir has achieved patent for publication. With this brief introduction, I would like to invite our guest speaker to start our today's session. Welcome you, sir.
Thank you. I think it was, the introduction was very good, more than what I have achieved. So thank you for that. And I also wanted to thank the committee for inviting me to present our work in this international webinar. So I thank you all for having me. So I will proceed with my presentation for the sake of time. So shall I continue?
Yes, sir. Please, sir. Please.
Okay.
It's a pleasure.
Okay. Thank you. So on the top of all, of course, I wanted to appreciate the invitation to participate in this webinar. So my title of presentation will be regarding the challenges and opportunities for drug discovery in developing countries. And I wanted to present this case using drugs for cutaneous leishmaniasis. So as an introduction first, I would try to briefly introduce what cutaneous leishmaniasis is and the current status in Ethiopia. So cutaneous leishmaniasis is the top 10 neglected tropical disease And this is one of the diseases difficult to treat because it's caused by more than 20 species, protozoal species belonging to the genus Leishmania. There are different forms of the Leishmania, visceral Leishmaniasis, which majorly affect the internal organs, but the most common form of Leishmaniasis is cutaneous Leishmaniasis because it affects 600,000 to 1 million new infections every year. And this drug causes ulcer. It causes lifelong scar and disabilities, very serious disabilities, and there will be the corresponding stigma. From this, there are also different types of cutaneous Leishmaniasis. The most relevant one to Ethiopia is the mucocutaneous Leishmaniasis because together with Bolivia, Brazil, and Peru, those 4 countries constitute for the total greater than 90% of the mucocutaneous Leishmaniasis, which causes the partial or total destruction of this mucous membrane of the nose, mouth, and throat. So when you come to the case in Ethiopia, cutaneous Leishmaniasis which is caused by Leishmania ethiopica, is endemic to Ethiopia. That's why the parasite is named Ethiopica. This lesion is very different from lesions that are caused by other Leishmanias protozoas because this is atypical in that it commonly occurs on the face. It is more severe than other parasites, and the healing usually needs longer period of treatment than lesions caused by others like Donovani, major ammanozonosis, and other types of the protozoas. This drug now is a very growing health threat to Ethiopia because now around more than 30 million pupils are at risk of this infection, and there is increase in prevalence from time to time, and currently there is a recent outbreak to a region called Somali Zone in Ethiopia. So this drug is serious. But it's a surprise because this drug has been the persistent for pupils living in Ethiopia and Africa and developing countries for a very long period of time, but still pupils are suffering. And the major reason for this is because of the inadequate treatment available. So the current golden or the standard treatment for cutaneous Leishmaniasis are antimonial compounds, as in sodium stevogluconate and meglumine antimonates, but those drugs have been used for more than 70 years. And because of that, nowadays there is a rapid surge in emergence of resistance. Those antimonial compounds are also well known for their serious adverse effects, which is usually complicated because the treatment, for them to be effective, prolonged treatment is required. So there is a very inadequacy regarding the current treatment. And if this is the case, there are also alternative treatment available. The first one is amphotericin B. It's available as a deoxycholate salt, and there is also a recently liposomal formulation of amphotericin B. But I hope you are familiar with amphotericin B because it acts on the cell membrane and it causes severe toxicity, and it's also very expensive. This is very important because Leishmaniasis commonly affect pupils residing in developing countries, and most of them have very low income, so it will be difficult for them to afford treatments like amphotericin B. There is also another alternative treatment, which is miltefosine. Miltefosine was an anti-cancer drug. However, it's approved for Leishmania in 2014. But this drug also is, though this drug is novel, there is some problems associated with it. First, it is teratogenic. So it's a very high-risk drug. It causes GI disturbance, and the other disturbing fact about this drug is it has low therapeutic window, which is exacerbated because this miltefosine has longer half-life. This drug also is very expensive, and its effect against old-world Leishmaniasis, which are Leishmaniasis common in Africa, its efficacy against those kind of Leishmaniasis is not well established. So this current available treatment, they show the available treatments are highly unmatched by the high burdens of cutaneous leishmaniasis in developing countries. And this is also getting even worse because of the rapid surge in resistance. So it's clear to see there is a very high unmet medical need in case of cutaneous leishmaniasis. So why is this the reason? The reason is because of the current trend there is in drug discovery and the resulting treatment imbalance. Today, so there is no safe and effective treatment for CL. And why it is very difficult to develop effective treatment is because developing treatment is a very lengthy and costly pharmaceutical innovation process. Because it's lengthy, it might take more than 20 years, and it's very costly. If we look at the recent estimate by Dimasi and its groups, its 2016 estimation, the average cost to develop a new drug is around 2.87 billion US dollar. But this seems a bit higher because there is a group known as Consumers Right Advocacy Group. This group used the same method used by Dimasi and this group, and they came up with a relatively low estimate of $1.4 billion. But regardless of these debates, the cost remains substantial in that developing a drug is very costly process. So because of this cost, and pharmaceutical firms are for-profit organization, they require very good financial return for the drug they develop. Not only for the drug they develop, but they also need the approved drugs to cover the cost of other drugs failed during this lengthy pharmaceutical drug discovery development process. So because of this, they channel their effort to develop drugs to diseases of high relevance to developed countries because of the lucrative market there for them to purchase their new drug. So this market-driven approach will force the current trend in drug discovery to diseases of high importance to developing countries, creating a very fatal imbalance because this imbalance is killing thousands of pupils in developing countries. And we can see the reason why, because if we look at the per capita income in United States and Norway, it is more than 7,000 US dollar per year per person. And in Switzerland, it is as high as 6,000 US dollar per person. But when we come to the other end of the tunnel, like 31 member states of the WHO in developing countries spend less than 35 US dollar per person per year, which get even worse in 4 countries, which are able to spend only less than $10 per year. So this shows the disparity in terms of market in between developing countries and peoples living in the developed world. So pharmaceutical companies cannot develop the drug for developing countries because of this difference in market. And the governments also in developing countries cannot have enough fund to develop drug. For example, we can see the 2016-17 total annual budget of Ethiopia, which was around 30 billion US dollar. So if we use this total budget, Ethiopia can only develop 5 drugs per year. Considering how many neglected diseases are there, it will be impossible. And if we go to even the health budgets from this $30 billion, the health budget is almost negligible in order to develop a drug for endemic traits in Ethiopia. So this has created an entire dependence on pharmaceutical companies to develop drug for diseases of Ethiopia and other developing countries. But on the other hand, pharmaceutical companies have low incentive to develop drug for diseases of highly relevance to developing countries. So the trend now there is in order to stimulate or to do some incentive to pharmaceutical industries in order to develop drugs for diseases known as neglected tropical diseases. So I try to put those 2 measures taken, the current measures taken in order to stimulate drug development by pharmaceutical companies in developing countries to for diseases which are prevalent in developing countries. One of them is called priority review vouchers. So this PRV was started by the Congress of United States in 2007. So they passed a bill in order to encourage drug development for diseases like tropical diseases, for rare pediatric diseases, and for medical countermeasures. And the main assumption here is the main impediment that is not leading to drugs for those diseases is the lack of financial incentive rather than the lack of scientific knowledge. So they propose this bill that will reduce the time to market by 4 to 6 months. That will give more competitive advantage for the voucher holders or for the developing companies. That will also generate greater financial return, and most importantly, it will help the patient sooner. So in this line, PRV was introduced in 2007. But nowadays, the impact of PRV is being highly questioned because currently the value, this priority review voucher value, its market value has dropped from 350 million, which was in 2016, to just 81 million in 2018. And in generally, the value less than $100 million is not considered attractive for pharmaceutical firms. And the other one is the developing country has no obligation to develop drugs which is affordable for the patient. The company can sell the drug at the cost desired. So because of this factor, PRV is no longer considered as an effective strategy. The other, which currently is considered more contributing to disease of developing countries, is PDP, also known as Product Development Partnership. So this PDP is a subcategory of PPP. PPP is Public-Private Partnership. So this Public-Private Partnership is the global non-profit alliance that is that wants to generate the necessary financial support in order to develop drugs, vaccines, and diagnostic for neglected tropical diseases. So there is some success story with PDP. I think some products are the testament for the early bird success of the PDP. For example, we can consider of paromomycin. Paromomycin for visceral leishmaniasis was developed in 2006 by One World Health Institute. And we can also consider some of the effects or products produced or they are being produced by the NDI. The NDI is the Drug for Neglected Disease Initiative. For example, we can consider fexenindazole. Fexenindazole is a drug which is approved in 2009 for human African trypanosomiasis, and currently this drug is as equipotent as the recently approved miltefosine for visceral leishmaniasis. So this drug is now undergoing a clinical trial for cutaneous leishmaniasis and visceral leishmaniasis as well. The other drug that's currently being produced by the NDI is topical amphotericin B preparation for cutaneous leishmaniasis, so that we can lower the toxicity by topical administration. There is other also studies which consider the combination therapy of imiquimod with antimony compounds, and there is also another combination for cutaneous leishmaniasis which combine miltefosine with paromomycin. So PDP is playing its good role. However, if we look at the total expenditure, for example, the 14%, which is worthy like $506 million of this global R&D fund, which was allocated for neglected disease in 2007, this $506 million went to through PDP in order to develop drug for neglected disease. However, some amount was able to be traced, which was around 145 million and higher. So from this traced expenditure, 48.2% went to private organization in developed countries. And if we look at the detail, 20.9% went to research contract organization. The rest 18% went to small pharmaceutical industries, and the rest 9.3% went to big pharmaceutical industries. And the others, institutes in developing countries, which are the academia and public research institutes, they receive an additional 22.1% of the total fund allocated. So this shows that from the total allocation, 70% went to institutes in developing countries. And if we look at the allocated budget for organization in developing countries, only 12.3% of the total allocation went to institutes in developing countries, where most of the diseases are primarily residing at. So this shows the contribution from the developing countries are being overlooked by the current paradigm in PDP and other neglected drugs initiative. So we believe those PRV and PDPs are still very important, but however, a change is important. One of the major reasons why the change is important, because the treatment produced so far has been not satisfactory. In fact, they are inadequate, because for more than 7 decades and more, no effective drug was developed for cutaneous leishmaniasis. And the other one in the current paradigm is assigning the scientists which are residing in developing countries where there is a high disease burden of this tropical disease. I think in order to produce effective and sustainable solution, we must consider the contribution of these scientists, of the local scientists. And the other one is the ongoing dependence. If we continue with this PRV and PDP measure, then the dependence on developing countries will continue. And I don't think this is the right measure, because I may use this Chinese proverb. It may not be appropriate, but they have a saying that don't give me fish, rather show me how to catch one. So if we doesn't find a way to empower research organizations and researchers in developing countries to be able to develop a solution for their problem, rather try to give them one, then I don't think we will solve the problem permanently. And the other one is, if we continue with this PRV and PDP measures, then the endogenous resources in developing countries which would be neglected. But we don't think this would be the right one, because as you see some of the drugs on the right of this yin-yang puzzle, it shows 2 drugs. The light one was pentanostrol. So this drug is now currently undergoing preclinical trials, undergoing formulation in another study. This was obtained from a plant which was being used by the Mayan traditional healers in Mexico. So from this plant, this pentanostrol was isolated, and it was found to be really active. And it's now, after some years, it's expected to enter into the cutaneous leishmaniasis drug pipeline. And the other drug is 2-hydroxyflavonone. This was also isolated to be highly active against antimony resistance Leishmania parasites. So this shows that we need to continue working on endogenous resources available so that we can avail the drugs like of this 2-hydroxyflavonone and pentanostrol. So we believe strengthening the local research capacity is a very effective and permanent solution if we want to tackle neglected tropical diseases. But it can be done together with the current measures, PRV and PDP, so that the collaboration by these 2 methods, even if they seem contrary, however, if we combine those methods, then they can be complemented one another in order to once and for all solve this morbidity and mortality associated with neglected tropical disease. So what is the challenge? Why countries, developing countries, are not able to develop solution is because the first thing is we mentioned earlier, that they have very low financial capability. So they are entirely dependent on on pharmaceutical firms in developing countries. So this is one of the reasons why disease of poverty will continue to be unabated if we went through this pipeline. So what are the current reasons for this dependence? Is the first one is there is chronic underinvestment in universities and research institutions, because as I mentioned earlier, the healthcare budget for research in developing countries is almost negligible compared to the demand that drug discovery needs. And the other one is there are very few capable institutions in developing countries. This is becoming a critical problem, because now developed countries help researchers in developing countries through scholarship and fellowship program. However, those trained doesn't have a tendency to return to developing countries. So this is creating or this is promoting brain drain. And the reason is because in developing countries, there is more potential opportunities, unlike developing country where this career pathway is not available because of the very few research capacity, research institutions available. So people tend to stay in developing countries. And the other important is if a researcher is motivated to develop a drug for neglected tropical disease, then developing countries currently are the right place, because all those DNDi, MMV, and others are currently or majorly located in developing countries. So we need to create more institutions in developing world. Other one is political instability and incoherent research initiative. Because of this political instability, one government may come and support some strategy like prevention, then after another one came and it may focus on technology transfer. It keeps going. So there is incoherent initiation when it comes to drug development for neglected disease, and it's one of the problems. The others is there is restrictive rules and regulation. Developing countries, it's a manifestation of developing countries, especially even if you want to purchase something, the rules and regulations are they're unbearable or it's difficult to overcome. And the other one is in developing countries, there is also a problem of disseminating the already done researches and to utilize them for decision making and other like. So this is some of the challenge we are able to identify. And why we need to work on developing countries, because there must be some opportunities to work. The first one, I believe, is the motivation of the researchers, because even if researchers in developing countries have very low wage and they may need additional jobs to make ends meet, however, this researcher are highly driven in order to develop treatment for disease which is highly relevant to the society they are a member or they currently live in. So this motivation should not be underestimated. And the other and most important additional thing is cost efficiency, because there is low wage in developing countries for researchers. So drug development is going to be less or cost effective in developing country than developing drugs in the developed world. And the other one is a sustainable solution I mentioned earlier, because if we have enough research institution, then we can continue to train the junior professionals, so that there will always be professionals and institutions that are capable of overcoming the challenge, the current challenge, and the challenge to come in the future. So it may create a sustainable solution to disease of the developing world. So these are some of the two opportunities we have in developing countries. And the other and very important one is the untapped biodiversity and biouniqueness in developing countries. I will try to explain this later, but in developing countries, especially those affected by neglected disease, they have rich biodiversity and biouniqueness. And we know that there are lots of novel compounds to be found from this rich biodiversity in developing countries. So this would be not only effective for NTD, but also for some treatment. Who knows, if there is a possibility we can identify a drug that can capable of curbing the morbidity and mortality associated with cancer. So we need to work on developing opportunities we have in developing countries. So what measures we need to take in order to explore this opportunity available in developing world? The first one and the most important is investing and empowering local researcher as the research and development actor for neglected disease. So the major problem with the current paradigm is it neglects the contribution of the developing countries. But as I mentioned earlier, researchers in developing countries are highly motivated to solve their own health threats. So if we provide them with the necessary finance and other support, then they will develop their own innovative solution rather than relying on pharmaceutical industries in developed world, which will try to develop only drug when they see it fits their own needs. So we need to empower local researchers in developing countries. The other one is, in addition to those primary stakeholders, we need other competent research stakeholders. For example, one of the limitations we have in developing countries is suppliers of those instruments, chemicals, and reagents that are very important. So even if one secured the constrained financial problems, then it may be difficult to find the appropriate research materials because of those few suppliers available in developing countries. We also need some change from the side of the government. As I mentioned earlier, rules and regulations are very prohibitive in developing countries, so the government needs to sharpen their rule in order to favor research for endemic health threats. And the other one is the government must try to make their decision based on results, research results. We believe this will be a best intervention in order to solve some of the issues we have in developing countries. So with this one, there are various measures to focus on, but I would try to discuss only natural product and clinical research. So as I mentioned earlier, the problem we have with current treatments for leishmaniasis is most of them have been used for a long period of time, so their structure is not new. So there will be a continuous development in resistance. So this is the first thing we need to tackle. The other one is both antimony compounds, amphotericin, and also the recently approved miltefosine. They are also toxic, so we need agents which are less toxic. And all the other measures or the current treatments available, they are costly. So we need affordable treatments as well. So these are the three major things we need to overcome, which are novelty, toxicity, and treatment affordability. So in this regard, we believe natural products could be the perfect source in order to overcome these three major problems associated with currently available treatments. We'll try to see one by one, but in order to headlight about natural products, since antiquity, natural products have served the human race in order to avail treatments for their major health problem. Of course, now in pharmaceutical company, there is a tendency to use high throughput screening and combinatorial chemistry. However, the impact of natural products in the current drug pipeline is still intact, because we can see the survey done, for example, as an example, for anti-cancer drug developed between 1940s and 2040, and it shows 50% of the drug are of natural products or semi-synthetic origin. So this shows the current impact of natural product is very significant, so we need to focus on those natural products, especially for cutaneous leishmaniasis. The reason is because natural products, they are low cost. Most of the natural products, they are low cost. And we have mentioned earlier, one of the major problems we have is affordability, because most people affected by NTD have very low income, and the treatments, on the other hand, are costly. So we need plans in order to reduce the cost. And the other one is safety. We know natural products are safe. Various studies have shown that one. And one of the major problems associated with current available treatment, like amphotericin, antimonial compounds, and miltefosine is toxic. So in this regard, plants could be a very good alternative. And the other one is novelty. We know that herbal products have evolved over millions of years, and they have very diverse in biological activity. Even some of them, their structure is so unique that medicinal chemistries won't be able to envision this kind of structure. So we need to explore more herbal products so that we would get more unique compounds. So more unique compound means a novel mechanism of action, so which would be very important in addressing resistance problem associated with the current treatment. So because of this low cost, safety, and ability to overcome resistance, we believe natural compounds are the perfect fit. And this is also shown by some of the studies. There are various studies that showed numerous plant extracts which shown potent activity against antimony resistant Leishmania parasites. And they also have low incidence of adverse effect, and most of them have low cost. So there is evidence of their importance in regarding to overcome the current challenge of this anti-malarial, anti-leishmanial treatment. So we need to work more on natural products. And this is very important. We believe it is a very colossal opportunity yet to be explored, because current in general in the world there are 250 up to 50,000 plant species. However, only 6 of them have been screened for their biological activity so far. So which means there is lots of plants to be screened and lots of novel structure to be explored. So this is very important. And the other one is when you come to the countries affected like Africa and others, there is a very grand biodiversity and a very high number of endemic plants. For example, if we see Africa alone, 25% of the world biodiversity is found in Africa. So it shows if we work on developing countries, then there is a very high potential to develop low-cost, highly safe, and novel anti-leishmanial treatments. And the other one is, aside working on developing novel treatments, in Africa, there is a very high tradition of using herbal medicine. Like 80% of the population, they rely on traditional treatment as their basic treatment needs. So, and also there is the currently available treatment have high cost and they require lengthy treatment. So if we do some clinical research and show statistical significance in terms of their efficacy and testing, then rather than developing agents from natural products, there is an opportunity to develop and use natural products as they are. So this is a colossal opportunity that needs to be considered in the future. So we can develop herbal products if we work on the clinical research for these herbal products available in developing world. The other thing that we believe will strengthen the local research capacity are clinical research. One of the bottlenecks for health innovation in developing countries is the insufficient clinical trial we have. Because clinical trials, as we all know, they are highly resource intensive. The most expensive stage of drug development are clinical trials. So this is one of the reasons which we don't have enough in order to support clinical trials. So this is one of the reasons why we have insufficient clinical trials. So efforts needs to be done in this regard. The other important thing is ethical and regulatory procedure. We all know ethics is the very underlying factor in clinical trial. So we need to have our very good ethical and regulatory procedure. Of course, as I mentioned earlier, there is a lack of research material infrastructure in developing country and operational obstacles like purchase, availing of resources and others. So we need to resolve these limiting factors in order to increase the clinical research in Africa and other developing countries as well. So, however, now this trend is changing, and nowadays Africa is emerging as an important destination in clinical trial, although this is underrepresented. And the major factor now why pharmaceutical companies are establishing clinical centers in Africa is there is a diverse population, which is very important in order to get statistical significance, efficacy against different world populations. So this is one of the factors, and of course, the other one is there will be reduced cost and time in order to recruit volunteers to be enrolled in clinical trials. So if we consider Asia, Latin America, Africa in general, 40% of all clinical trials in the world are currently being done in developing world. However, the major problem is most of these clinical trials are addressed to diseases of high importance to developed world. So what we need to do is to resolve these financial resources and of course focus on diseases of high importance to developing world rather than diseases of only on diseases of the developed world. So we need to promote clinical trials because they will undoubtedly raise the research standard we have in developing countries, and most importantly, since they are resource intensive, they will bring the badly needed investment to do research relevant to health rates in developing world. So a study have shown that developing capacities through education and training in diagnosis, clinical management, clinical care and data management, they have improved the clinical research in Africa and other developing countries. So in this, to do our part, CDT Africa is currently offering postgraduate and fellowships in clinical trials so that we can fill the gap of professionals required for clinical trials in Africa. And of course, our major intention is, as I mentioned earlier, we have various natural products with safety, efficacy, and novel compounds. So the only thing left to do is to do clinical trials in order to show significant numbers, significant efficacy and toxicity for those data so that we can use them as they are with some formulation study. So we are currently really working on developing novel natural products to use them as they are for clinical, for cutaneous leishmaniasis and other topical diseases like TB and malaria. So just to conclude and to do some recommendations. So the slanted hedge, it shows the current paradigm that the most of treatment goes to through PRV and PDP, and unlike the population, the researchers and the research institution available we have in developing countries is very low. So if we want to restore this balance, we need to go to the horizontal line where there will be a contribution from PRV and PDP, and at the same time, we need to build local research capacity so that we would increase the number of research institutions available. We can increase the number of professionals we have in developing countries. We can get drugs like of 2-hydroxyflavone and pentanostrol, and we can also develop herbal products so that we can have sufficient treatment for diseases of neglected tropical diseases. So you can get this research work is currently is accepted by SS Medicinal Chemistry later. So if you want to read the whole paper, you can go to the ahead of print section of SS Medicinal Chemistry, and you can get the whole work free of charge. So thank you for your attendance. And if you have any questions or comments, then
thank you very much, sir, for your wonderful and informative lecture on the topic challenges and opportunities for drug discovery in the developing countries. And also, we all thank you once again for letting us to know about the things which are being faced, the lack of development of clinical trials in the developing countries like Africa, as well as how to strengthen this local health research capacity in the developing countries. Thank you very much, sir, again for your wonderful session.
Thank you.
Now it's my privilege to welcome the Director of Sri Padmavati Educational Institutions, the patron of our today's program, Sri P. Praneeth sir, to the program. Sir, we welcome you, sir, to the valedictory program.
Welcome.
Sir, I heartily welcome you to the valedictory program of this Indo-African webinar series, sir. Director of Sri Padmavati Educational Institutions, Sri P. Praneeth sir. We welcome you, sir, to the today's session.
Hello, Dr. Abdrrahman. Please wait for a moment. He is our director and today's patron
No
for
problem.
this international webinar series. So we would like
No
to
problem.
introduce him to you.
We welcome you, sir, to the today's program.
Yes, thank
Good afternoon,
thank
sir.
you, madam. Very good afternoon and very good afternoon. Yeah, very good afternoon to team and dear speakers. Yeah, I think there's a small problem with my connectivity. I'm extremely sorry for that.
No issues, sir. So today's speaker is Dr. Abdrrahman Shemsu Surur. And
Yes, sir,
speaker,
fine.
Dr. Abdrrahman, he is Mr. Praneeth Penimadu, the patron of International Webinar Series, and he is the director of the Padmavati Educational Institutions.
Yeah, hello, sir. Very good afternoon.
Sir,
It's great meeting you virtually.
Yes, sir, we welcome you, sir. Sir, now I would like to, we came to
Yeah,
the
fine.
end of this Indo-African webinar series, sir. Part 1 and Part 2 was conducted
Fine.
on 29 September and 30th September, sir. And today is the last
Yes, yes.
part, which is based on the theme challenges and opportunities for drug discovery in the developing countries, sir. Now I would like to request our beloved director, Sri P. Praneeth, sir, to address the virtual gathering.
Very good afternoon, dear team, the Padmavati School of Pharmacy, faculty members, students, and today's speaker of the day, Dr. Abdrrahman, on the webinar topic challenges and opportunities for drug discovery in developing countries. That's Indo-African International Webinars. Actually, that's very great to be a part of the webinars, and also I've been hearing sir's speech also for the past 15 minutes. That was really great. And thank you, sir, for sharing your knowledge to us and sharing the updates. And usually this, as I told earlier, that previously it was very difficult to conduct national and international webinars because it was very difficult for us to get the speakers' dates and for them to bring down to our campus. All these issues were there. But now I think due to this pandemic, I think we should feel that really that is an biggest gift for us, where speakers could spend their time through the online and where they can share their views, thoughts, and update our knowledge with a great experience. Really, pandemic, that is COVID-19, has brought many changes in the education system, and that has bring a positive changes. That's what I call, where I think we are able to get in touch with great personalities, great speakers, and it's becoming easy for us to get that is the sharing of knowledge. So that is becoming very easy. So I think we should thank COVID for that. So where it's making easy for us, and on behalf of the management, I thank and congratulate Sri Padmavati School of Pharmacy team for conducting such kind of international webinars for the benefit of the students, staff, for the updation of the knowledge. And I also thank the speakers who have been there. And I also extremely thank today's speaker, Dr. Abdrrahman Sir, for his wonderful speech and for wonderful sharing of knowledge to all our students and staff. Thank you, sir. From the behalf of management, I thank you. I thanks and congratulate the complete team for the successful completion, conducting and the completion of webinars. Thank you. Thanks a lot.
Thank you very much, sir. Sir, once again, thank you very much for encouraging and supporting us to do this kind of webinars. Thank you, sir. Now, I would like to request our organizing secretary, head of the Department of Pharmaceutical Analysis, to present the closing remarks for this today's session. Dr. K.K. Rashekar, sir.
So, distinguished webinar panel members, participants, ladies and gentlemen, good morning, good afternoon, good evening to all. Today's session is the third and last part of the Indo-African International Webinar Series 1. And the first two parts of this webinar series demonstrated the challenges of vaccine production and quality assurance. And in today's session, the challenges and opportunities for drug discovery in developing countries with an example of cutaneous Leishmaniasis was highlighted by esteemed speaker Dr. Abdrrahman Shemsu Surur. It is pathetic to note that cutaneous Leishmaniasis, one of the most prevalent and neglected diseases in developing countries of Africa, do not have a new effective drug developed in past 7 decades. Particularly, this mucocutaneous leishmaniasis, the stigmatic form of cutaneous leishmaniasis, results in partial or total destruction of mucous membranes of nose, mouth, and throat. This is very miserable and very pathetic. And people of developing or poor countries like Ethiopia and Peru suffer with mucocutaneous leishmaniasis. This is really miserable. It was clear today, as highlighted by esteemed speaker, why drug discovery and designing matters a lot and how vital it is for happy living of human race. From our speaker's perspective, we noted that the current trends in drug discovery and market-driven approach of pharmaceutical industries have cornered a few diseases and infestations prevalent in developing countries. Treatment imbalance is always there, and we have to visualize it as an open and new era to develop novel treatment regimen. Speaker also stressed about the complex policy making, political process, priority review vouchers, product development partnerships, natural product research, and other factors that stimulate the drug development against diseases neglected in developing countries. Therefore, it is important to promote clinical trials in developing countries for raising the research standards and bring the badly needed investments. Lastly, it is evident from today's lecture that the treatment imbalance exists and it may exist in future too, but many diseases are neglected by pharmaceutical industries in developing nations. This has to be noted with care, and this demands innovative strategies to strengthen the local and international health research. Thank you all for your patience. I wish you all a very good day and goodbye for this International Indo-African International Webinar Series 1. Thank you. Over to Kirtisika.
So thank you very much, sir, for highlighting the concepts which was delivered by our honorable speaker, Dr. Abdrrahman Shemsu Surur, sir. Now, as a part of gratitude from our institution, now I would like to request the convener of today's program and the principal of Sri Padmavati School of Pharmacy, Sri Dr. D. Ramnaiklu, sir, to present the certificate to our resource person, Dr. Abdrrahman Shemsu Surur, sir. Sir, 1 minute, sir.
Sorry, can I hear the last part?
Yeah, Abdrrahman, please wait. Please wait.
Sir, sir. I request Dr. Ranganaikulu sir to present the certificate of appreciation to our today's speaker, sir.
So it's very pleasant for us to hear that magnanimous lecture in this challenge and development of the vaccine, particularly this report of examples. Sir, is very much useful for the present crisis of this all African country facing with a lot of problem with this kind of pandemic invasion. Thank you very much. As a token of respect on behalf of this institute, I would like to request you to accept our token of appreciation certificate in this regard. I request the organizers to submit this. At the same time, I take this opportunity to convey my special thanks to the Pharmaceutical Analysis Department, Dr. Rashekar, the organizing secretary of this organization department, and Mr. Charan, and all the team, Ketishika, and all the members know very much strongly they have made this program a successful program. Thank you very much. On behalf of all this pharmaceutical analysis department, I request today our speaker, Abraham Shemsu Surur, to accept our program. Yes, sir. Thank you very much, sir. So, over to Charan.
Thank you.
Thank you, sir. Thank you, sir. Thank you very much, sir. Now, I would like to request the coordinator of this Indo-African International Webinar Series, Dr. A. Srinivas Sharan, sir, to present the vote of thanks for this event.
Thank you very much. Respected organizing committee members, honorable speakers, our most valued and invited guests, and all the virtual participants. It's my privilege to have been asked to propose a vote of thanks on this occasion. I, on behalf of Indo-African International Webinar Series 1 and the entire organizing committee, extend a very hearty vote of thanks to today's speaker, Dr. Abdrrahman Shemsu Surur, for gracing your important work and sharing with us your findings and opinions today on the topic Challenges and Opportunities in Drug Discovery in Developing Countries, the example of cutaneous leishmaniasis. Thank you very much, Abdrrahman sir. Thank you very much. A big thank you to Mr. Noor Ahmad Saedato, the speaker of Part 1 of IAIWS-1, and Dr. Muhammad Rafi Mokla, speaker of Part 2, for their efforts toward excellent demonstration of vaccine development throughout this international webinar series. So I thank all the three speakers for their valuable presentation in our webinar. I would also like to take this opportunity to place on record our hearty thanks to Srimati P. Salochana Madam Garu, Chief Patron of the webinar and Chairperson of Sri Padmavati Education Institutions, and Sri P. Pranit Garu, Patron of the webinar and Director, Sri Padmavati Education Institutions, for their continuous support and motivation in conducting this type of webinars. Thank you very much, Madam and Sir. Thank you very much. I also extend my thanks to Professor D. Ranganaikulu Garu, convener and principal of Sri Padmavati School of Pharmacy, and also to Dr. C. Sridhar Sir, co-convener of this webinar, for their enormous cooperation in the organization of this event. An event like this cannot happen overnight. The wheels started rolling weeks ago. It requires planning and a bird's eye for details. So we have been very fortunate enough to be backed by our organizing secretary, Dr. K.K. Rashekar sir, and a team of very motivated and dedicated colleagues, Srimati P. Kirtisika, Ms. J. Bhavita, Ms. H. Nilofar, and Srimati C. Harita, faculty of Department of Pharmaceutical Analysis, who know their job and they are always result oriented. We also thank all the faculty members of Sri Padmavati School of Pharmacy for their cooperation in conducting this webinar successfully. Last but not least, we express our sincere thanks to all the registered participants who have been part of this international webinar series, and we thank them very much for their active participation. We hope this webinar has enriched only your knowledge better and wiser. Thank you very much. Once again, we thank all of you for being with us this good evening. It's been a great pleasure to meet you all virtually. Thank you, one and all.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे